I'm Celine Hilda Mary going to present about the cross-matching. Introduction. Cross-matching is one of the most important serological procedure pertaining to blood group serology and is the fundamental procedure responsible for safe blood transfusion. Basically, cross-matching is an antigen-antibody reaction, a correct interpretation of which is the most essential preliminary step in the practice of safe transfusion of blood. By cross-matching, we are able to detect the atypical and clinically significant antibody, mostly IgM and IgG, present in recipient serum or in donor serum. Also, by auto-control, we are able to detect autoantibody in patient himself. Cross-match test is carried out to ensure that there are no antibodies present in patient's serum that will react with donor cells when transfused. Unless there is an urgent need for blood, a cross-match must be performed for red cell transfusion. Functions of cross-match. It is final check of ABO compatibility between the donor and patient. It may detect the presence of an antibody in patient's serum, which will react with an antigen on donor red cells. To ensure that patient or recipient is supplied with a compatible unit of antigen negative blood. To prevent hemolytic transfusion reaction and also to detect immunologic autoantigen and autoantibody, and blood to be issued has to be processed accordingly. All this done with sole aim for safe transfusion of blood. Next, types of cross-match. Cross-match testing procedure has been divided into two parts: major cross-match and minor cross-match. Major cross-match included the donor red cells and patient serum. That is, to the donor red cells, we have to add the patient serum. Maximum amount of donor red cells and minimum amount of patient serum has been taken. But in minor cross-match, patient cells are added with the donor serum. Next, what is the principle involved in the cross-matching? That is, the major cross-match is done to detect any serological incompatibility between donor cells and patient serum. But the minor cross-match is done to detect any serological incompatibility between patient cells and donor serum. Cross-match is verified with a Coombs reaction to detect even the incomplete antibodies present in the blood cells. Next, cross-match techniques. There are different techniques has been explained about the cross-matching. First type, immediate spin method. Second one, saline room temperature technique. Third one, indirect antiglobulin technique, that is Coombs test. And fourth one is the albumin addition technique. First we will see about the immediate spin technique or saline room temperature technique. Immediate spin technique or saline room temperature technique is enough to rule out any ABO grouping error, but this technique is inadequate for identification of clinically significant IgG type of antibodies. Both these techniques are not good because antibody screening has not been carried out in our country. Next, procedure. This is the procedure involved in the spin technique. That is, first we have to label the tubes as major and minor cross-match with donor number and patient ID. And next, using a micropipette, we have to add 1 volume of 2 to 5% of red cell suspension to the labeled tube, and 2 volume of serum to the same tube has been added. Next, we have to mix the tube well and centrifuge at 1000 rpm for 1 minute. And next, gently shake and observe for agglutination or hemolysis. No agglutination or hemolysis indicates the compatibility of the blood sample. If there is any agglutination or hemolysis occurs, that indicates the incompatibility of the sample. This is the pictorial representation of the immediate spin technique, that is, which helps to detect only IgM antibody which is reactive at 22 degrees centigrade, and which cannot be helps to detect the IgG antibody because which is reactive at 37 degrees centigrade, so which cannot be detected. This is the step. In the first step, we have to take the patient serum for 2 drops, and to this patient serum, next we have to add the 1 drop of 5% of the RBC cell suspension and mix it well, and say which is immediately centrifuged at the temperature of 22 degrees centigrade, and which is observed for the agglutination. If there is a agglutination, means that indicates the incompatibility. Here there is a agglutination is formed, so which is the incompatibility one, so we should not transfuse the blood from the donor to the patient. That's all about the spin technique. Next, we'll move on to the indirect anti-globulin technique. That is IAT test is widely used in cross-matching as it detects majority of incomplete antibodies. Next procedure: put 2 drops of serum in pre-labeled tube and 1 drop of 2 to 5% suspension of red cells, which is incubated for 45 to 60 minutes at 37 degrees centigrade. Next, gently shake and observe for agglutination or hemolysis. If test tube show any agglutination or hemolysis, that means incompatible at 37 degrees centigrade. If no agglutination or hemolysis is present, wash the cells 3 to 4 times with saline and decant the last wash completely. Next, add 2 drops of anti-human globulin reagent, that is Coombs reagent, to the test tube. Next, gently shake and read the result immediately. If no reaction, wait for 5 minutes. And next, centrifuge at 1000 RPM for 1 minute. Shake gently and observe for agglutination or hemolysis with optical aid. If no agglutination or hemolysis means which is a compatible one, but if there is any agglutination or hemolysis occurs which is a incompatible one, so we should not transfuse the blood from donor to the patient. If the test is negative, add 1 drop control IgG coated or red cells. Centrifuge again at 1000 RPM for 1 minute and observe for the hemolysis or agglutination. If no agglutination or hemolysis, the test is invalid and repeat the procedure. It's the interpretation. Hemolysis or agglutination at any stage of test procedure except after adding control IgG coated red cells indicates incompatibility of the blood cells. This is the pictorial representation of the anti-globulin technique. To the 2 drops of the patient serum, we have to add 1 drop of 5% RBC, donor RBC cells, and incubate this mixture for 37 degrees centigrade at 1 hour. And you have to wash the sample for 3 times. And next we have to add the 2 drops of AHG reagent, that is anti-human globulin reagent, and mix it well And next you have to centrifuge the mixture and view for the agglutination. If there is no agglutination, means which can be compatible one, but if there is agglutination or hemolysis occurs, which is a incompatible one. So we should not transfuse the blood from donor to recipient. Next we will move on to the factors affecting IAAT, that is temperature at 37 degrees centigrade, serum and cell ratio is the another factor. Incubation time also affect the IAAT technique, and suspending medium such as sensitivity of IAT increased with the addition of 22% bovine albumin enzymes or LISs. These are the factors which affect the IAT technique. Next, we'll move on to the albumin addition technique. In this technique, put 2 drops of serum and 1 drop of 2 to 5% red cell suspension in a pre-labeled test tube. Gently shake it well. And next we have to add 2 drops of bovine serum albumin and gently shake the test tube, which is incubated for 30 minutes at the temperature of 37 degrees centigrade, and centrifuge the test tube for at 1000 rpm for 1 minute. Next, wash the test tube for 3 times. Minute amount of human protein can be neutralized. AHG ensures that the saline is completely decayed after each wash. Next, add 2 drops of AHG to the dry cell button. Gently shake and read the results immediately. If no reaction, wait for 5 minutes. Again, centrifuge the tube at 1000 rpm for 1 minute. Shake gently and absorb for agglutination or hemolysis with optical aid. No agglutination or hemolysis occurs, that is the compatible one. If there is a agglutination or hemolysis seen, which is a incompatible one, so we should not make the transfusion from patient to recipient. If the test is negative, add 1 drop control IgG coated red cells. Centrifuge again at 1000 rpm for 1 minute. And next we have to absorb for the hemolysis or agglutination. If no agglutination or hemolysis occurs, the test is invalid and repeat the procedure. This is the pictorial representation of albumin addition technique. Here also we have to take the 2 drops of the patient serum and add 1 drop of the donor RBC, mix it well, and next you have to add 2 drops of BSA, which is incubated at 37 degrees centigrade for 1 hour and which is washed for 3 times, and again AHG sample is mixed, which is centrifuged and view for absorbed for the agglutination. If there is no agglutination, means a compatible one, and there is agglutination which indicates the incompatible one. Next we will move on to the causes of positive results in cross match. First point, incorrect ABO grouping of patient or donor. And next one, an allo antibody of the patient reacting with the corresponding antigens on donor cells. An autoantibody in patient serums reacting with corresponding antigen on donor red cells. This can be solved by putting autocontrol, which will be positive. Donor red cells with a positive DAT And next, problems in patients serum, example multiple myeloma. And last factor is the dirty glassware. These are the causes of the positive results in the cross matching test. And what are the drawbacks of the cross matching test? They are included: RH typing errors cannot be detected by cross matching. Next, it can detect only antibody specific for red cell antigen of the donor. A donor unit without appropriate antigen and with very weak antigen may thus fail to detect the corresponding antibody even if it's present in serum of the patient. Next, present method of compatibility testing cannot detect any antibodies to either leukocyte antigen or platelet antigen. Thank you.